Hey guys, so today we're going to be taking a look at the genetics of organisms, and we're specifically going to look at the inheritance patterns and Mendelian genetics. So let's get right into it. As an overview, we're going to be looking at alleles, Mendel's laws, genotype versus phenotype, different kinds of zygosity, Punnett squares and how we can apply those, autosomal and sex-linked inheritance, monohybrid and dihybrid crosses, which is really the application of all of this together, and then chi-squared tests, which will be used kind of in an experimental setting. So to start off, we'll look at alleles here. And primarily, one of the things that you should know is that most of the cells in your body are called autosomal cells, and those are diploid. So when a cell is diploid, it has two chromosomes, and one of those is from your mom, and one of those is from your dad. So each chromosome has one allele. An allele is basically a version of a gene. So for this example here, let's say you received this chromosome from your dad and this chromosome from your mom. So your dad gave you a big A allele and your mom gave you a little A allele. So let's say this is hair color. So if your dad gave you brown hair and brown hair is a big A, and your mom gave you a dark hair gene and that was a little a, you'd have a big A and a little a for yourself. Now this kind of brings into question dominance versus recessiveness here. And a dominant allele is one that will show. So let's say you have a dominant and a recessive allele. The dominant one is the one that's going to be expressed and that you're going to show externally. So in that case, let's say dark hair was dominant over lighter hair, your dark hair would show. And then this is kind of what genotype and phenotype are. So your genotype is going to be the actual set of genes and the set of alleles that you have. So in this case, we were referring to, if we got the big A and little a, your genotype would be big A, little a. Whereas your phenotype is going to be the actual physical trait that you express. So that would be dark hair, for example. And your phenotype is actually dependent on your genotype, as you kind of saw there. So yeah, again, this is just explaining it a little bit more here. And we have a physical example. So in this case, we're looking at red and blue flamingos, where the big A would make a red flamingo and the little a would make a blue flamingo. So here, when we have two big A's, we're going to get red as our phenotype. When we have a big A and a little a, the big A is dominant, so we're still going to get red as our phenotype, even though we have a different genotype than we did in the first case. And then for the homozygous recessive, this is two little a's, then we are going to get the blue phenotype. So now let's look into Mendel's laws of inheritance. And there's three main ones here. Gregor Mendel was basically the father of all inheritance, which is all I really want you to know for that. So we'll start off with the law of dominance here, and this is, again, going into the dominance and recessiveness. So if you have one dominant allele and one recessive allele, you're going to have a dominant phenotype. So again, if we use the flamingos for an example, you have one red allele, which is the dominant, one blue allele, which is the recessive, you're going to have a red phenotype. Then the law of segregation says that alleles separate during formation of gametes. And gametes are, as you remember, sperm and ova, so the eggs. And basically what happens is when you create your sperm or your ova, you're going to have one allele from your mom and one allele from your dad, right? Well, each of these alleles get split. So then you're going to have half of your gametes with your mom's allele that they gave you, and then half of your gametes with the dad's allele. Now, for the law of independence, this says that the way alleles separate during gamete formation is independent of all other alleles from other genes. So what this is really saying is that the gene that you have for hair color and the gene that you have for eye color, let's say, they'll be independent of each other. So it doesn't matter what your gene is there, they'll separate independently, and there will just be a randomization between the two for what happens and what you end up expressing. And of course, there are exceptions to this because, as you can imagine, some genes are actually on the same chromosome, so those would not fully be independent together. But this was that was discovered after Gregor Mendel made these laws. So we've already described a little bit of homozygosity and heterozygosity. Homozygosity can be both when you have two of the dominant alleles or two of the recessive alleles, and then heterozygosity is always going to be two different alleles. Now, for Punnett squares, these are really just the tool that we use to actually figure out the inheritance pattern. So if we have a genotype of two parents, then we can figure out the probabilities and the ratios of the genotypes and their offspring. So in this case here, we're looking at parents both with a big A and a little a. So they're heterozygous for this gene. And right now we're considering the big A allele would make a green pea, and then the little A allele would make a yellow pea. So for each parent, we would put the big A here and the little a there. And then we make a little square kind of underneath and beside them. And now we just kind of go across. So we have a big A from here and a big A from here, and we get two big A's for this one. Now let's go over here. So we have a little a here and a big A from here. We get a big A and a little a. Down here, same thing, big A, little a, we get a big A and a little a. And then for the last one, we have two small a's, one from each parent. And this is the only one that we get the yellow phenotype, because we have two recessive alleles. So now we'll look at autosomal versus sex-linked inheritance. And autosomal is really just your regular cells, so it's just anything that is not sex-linked, so just regular inheritance. Whereas sex-linked, on the other hand, is when the gene that we're talking about is on the X or the Y chromosome. And as you remember, males have an X and a Y chromosome, and females have two X chromosomes. So as you can see, the inheritance patterns are going to be different for males and females in this case if a gene is on one of those chromosomes. So here, let's get a nice look at it. So let's say the mother with the two X's and the father with the X and the Y. The daughter is going to get one of her X's from the mother, and then is going to get the only X from the father. And this is really key to understanding the inheritance pattern. So no matter what, one of the female's X's is going to be the father's only X. Whereas for the male child, he is guaranteed going to get the Y from the father because the mother does not have a Y. And then the X that the male child has, is going to be one of the two X's that the mother had. Now let's look into the application of all this a little bit. So a monohybrid cross is just about one trait. And it's called a monohybrid cross because it's usually considering the cross of two heterozygous parents. But it's about the process though. So we usually use these kind of crosses in order to find the genotype of the P generation. So this is, it's just purely investigative for the most part. So we have a few terms that we like to use for this. So the P generation is the parental generation. And for this is usually when you breed two homozygous parents. One will be fully recessive, as in right here, and then one will be fully dominant, like this one right here. And then the result of this is going to be all heterozygous offspring, as you can see here, because all of them are going to have a big G and a little g. Whereas the F2 generation is when you take all of these heterozygous offspring, you get rid of the parents from whatever population you're using, and then you breed these guys together. So then you're going to have your big G, little g crossing with your big G, little g. And the result of this is going to be the same that we saw in the previous example, where it was you're going to have three dominant phenotypes here and then one recessive phenotype. So that's where we get the 3-to-1 phenotypic ratio, and this is very characteristic of monohybrid crosses. Now, dihybrid crosses are a little bit more complicated, and it is what it is. It's just two traits instead of the one trait that we're looking at. And again, purpose is very similar. It's often where you're just trying to figure out the genotype of the parents based off of the ratios of offspring that they give. So our P generation here, we're considering two traits. So here our B alleles are one trait and our C alleles are another trait. So this one that I'm circling right now has two dominant B alleles and then 2 dominant C alleles, whereas this one is both recessive there. So our P generation, we're going to cross fully homozygous parents, and one is going to be fully dominant and the other is going to be fully recessive. And same thing, like in the past, the result of that is going to be all heterozygous offspring in the F1 generation. Now, we remove the parents, and then we cross only the heterozygous offspring here to get the F2 generation. And from here, we're going to get a 9:3:1 ratio. And what the 9:3:1 ratio is explaining the 4 different kinds of phenotypes that you can get. So with the 9, so our first phenotype is going to be the 2 dominant phenotypes, which are in this case yellow base color and then the normal wings. So here we can just count them up. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9. So some of them are homozygous for some traits, others are heterozygous, but no matter what, they're going to be expressing the dominant phenotype here. Now the 3, we have, for example, we'll start off with the yellow and then the clear wing. So we have 1, 2, and 3. Now all of these have the double small c, right? That's how we're getting that different phenotype. And then for the second 3, we have these 3 right here because they have the white base color, but they have normal wings. And then the last little one right here is from the double homozygous recessive, and you only get one of these, 1 out of 16 of all of them together. Now, the chi-squared test. We'll go into this a little bit because you'll be using this in the lab component that will be in the next video. So this is really just a way of determining if your results are actually due to the phenomenon that you think they are due to. So, and it basically gives you a probability at the end, and it says if the probability is really, really low, which is going to be your p-value, if it's under 0.05, then it basically says that what you're trying to characterize is not due to chance. It is due to the phenomenon you're considering. So we'll go through the steps here first. And number 1 is to figure out what your prediction is. So what your null hypothesis is. And if the chi-squared test happens to be a p-value over 0.05, then that means that it's going to be due to chance and that it won't fit the hypothesis. And then the next step would be to make a Punnett square and figure out all the probabilities of each outcome. So that would be like this if it's a dihybrid cross, or like this if it's a monohybrid cross. And then your probabilities are basically just, in this case it would be 9 out of 16 for this one, 3 out of 16, 3 out of 16, 1 out of 16. Whereas for this one, you're going to have just out of 4. And then we are going to use the chi-square formula based off of the probabilities that we came up with here. And for this one, this means chi-squared here. And then it equals to the sum of, which is this little character here, the observed number of outcomes for that particular phenotype, minus the expected number of outcomes, which is going to be based off of a ratio and the total number that were in the sample. You square that together and then divide it by the expected. And then the degree of freedom is going to be needed to use to find the p-value afterwards. And the degree of freedom is basically just 1 less than the number of possible outcomes. So for a monohybrid cross, we only have 2 possible outcomes, right? We have the dominant phenotype or the recessive phenotype. So there's 2 possible outcomes, we have a degree of freedom of 1. Whereas the dihybrid cross, we have 4 possible outcomes, right? Because we have the 9:3:1, and we saw here from when we went through there. So 4 minus 1 is going to be a degree of freedom of 3. And then once you have all this together, you can use the chi-square table that will be provided to find the actual p-value. So let's go through an example of this because I know that it's a little bit difficult just to explain it as is. And we found a nice little example online. So here it's kind of showing, we're using p's again, and it shows the observed frequencies of the peas in the F2 generation here. And in total, we have 1,216 peas. Now, because this is a dihybrid cross, because we're looking at yellow versus green as one trait, and then wrinkled versus smooth as another trait, it's a dihybrid cross. So we're gonna look for that typical 9:3:3:1 ratio again. So the first thing that we want to do is figure out the number of expected peas that we should get that fall under each of the phenotypes here. So to do that, we have our total number of peas, right? And then what we would expect is for the smooth yellow peas, 9 out of 16 of the offspring should be smooth and yellow. Then 3 out of 16 of the offspring should be smooth and green. Same thing for the yellow wrinkled, and then only 1 out of 16 should be the ratio for the wrinkled green peas. So if we do the math a little bit, we just take this total here and literally just do the multiplication. You can divide this by 16 first to get this standard number, and then multiply it by 9 in this case here, by 3 to get the 2 middle ones, and then by 1 to get the last one. Then we put all of that into a table to keep ourselves organized here. So we have our observed number, 701, 204, 243, and 68. These are all just from this table here. And then our new expected numbers that we use from that equation right there. And we have the same total there. If you don't have the same total, then there was an issue with your math. Now, once we have that, we can start plunking them into the formula from a couple slides earlier. And the way it shows here, it just kind of gives a few extra steps just to make it a little bit easier on you. So it always helps to show your steps so we can kind of figure out where we went wrong and improve from that. So then basically the values that we get here from the formula, we have an individual one for each phenotype. These are the individual chi-squared values, but you're going to add them up in order to get the total value. And when we add those up, we get 4.76. Now, this is a table that's going to be given to you. This is just a predetermined thing. And here we only are highlighting the 3 degrees of freedom because, again, it was a dihybrid cross. There are 4 options. Subtract 1 from that, and you get the 3 degrees of freedom. And because our chi-squared value was 4.76 in total, that's between here and here, as you can see, right? So now we're looking at between 0.25 and 0.1. So that means that we have between a 10% chance and a 25% chance that the difference we saw between our observed and expected values are due to chance. So therefore, we can determine that our cross follows a regular dihybrid inheritance pattern and not any diverse phenomena. So in the next video, we're just going to be taking a look at some practice problems to start off. So we'll do a couple examples of some monohybrid and dihybrid crosses. And then we are going to do a virtual fly lab to be more specific. So make sure to review the content that we've looked at here, and next time we go look at the next video, we'll be good to go.